Transcriptional regulation. It is the process by which the expression of genes is controlled at the level of transcription, the synthesis of RNA from DNA. It involves the binding of regulatory proteins to specific regions of DNA, such as promoters and enhancers, to either activate or repress transcription. Here are some key mechanisms involved in the regulation of transcription. First is transcription factors. Transcription factors are proteins that bind to specific DNA sequences and regulate gene expression by either promoting or inhibiting transcription. They can be classified into two main types. One set is activators and second is repressors. Activators bind to enhancer regions and interact with the basal transcription machinery to enhance transcription, while repressors bind to silencer regions and interfere with the binding of activators or the basal transcription machinery, thereby inhibiting transcription. Second is the promoters and enhancers. Promoters are DNA sequences located upstream of genes that provide binding sites to the basal transcription machinery, including RNA polymerase, to initiate transcription. Enhancers are regulatory DNA sequences that can be located both upstream or downstream of the gene or even within introns. They contain binding sites for transcription factors, and their interaction with activators or repressors can modulate the rate of transcription initiation. Third is coactivators and copressors. Coactivators and copressors are proteins that do not directly bind to DNA but instead interact with transcription factors and other components of the transcriptional machinery. Coactivators enhance gene expression by facilitating the assembly of the PI initiation complex and recruiting additional transcription factors. Copressors, on the other hand, inhibit gene expression by interacting with repressors and modifying chromatin structure to prevent transcriptional activation. At number 5 is chromatin structure and epigenetic modifications. The accessibility of DNA to the transcriptional machinery is influenced by the structure of chromatin, which can be open or closed. Chromatin remodeling complexes and histone-modifying enzymes can modify the structure of chromatin by adding or removing chemical groups to histone proteins or DNA itself. These modifications, such as acetylation, methylation, and phosphorylation, can either facilitate or inhibit transcription by influencing the accessibility of the DNA to transcription factors and the transcriptional machinery. At number 5 is transcriptional coactivators and mediators. Transcriptional coactivators and mediators are multi-protein complexes that bridge the interaction between transcription factors and the basal transcription machinery. They facilitate the assembly of the PI initiation complex and help in the recruitment of RNA polymerase and other transcriptional components to the promoter region, thereby promoting transcription. And the last one is non-coding RNAs. Non-coding RNAs, such as microRNAs and long non-coding RNAs, can regulate transcription by binding to target mRNAs or genomic DNA and influencing their stability, translation, or chromatin structure. They can act as both activators and repressors of transcription, contributing to the fine-tuning of gene expression. Now have a look at the diagram. Activators bind to the enhancer region in the DNA. Then the DNA bending proteins brings the bounded activators to the promoter region. The activators bind to the transcription factors and mediator proteins, forming a transcription initiation complex. The complex promotes the binding of DNA polymerase to the promoter region, enhancing gene expression. Thank you for watching, and don't forget to subscribe, like and share.